To divide, a cell must complete several important tasks. It must grow, copy its genetic material, DNA, and physically split into two daughter cells. Cells perform these tasks in an organized, predictable series of steps that make up the cell cycle. Mitosis is the process by which a cell replicates its chromosomes and then segregates them, producing two identical nuclei. Mitosis is generally followed by equal division of the cell's content into two daughter cells that have identical genomes. In eukaryotic cells, the stages of the cell cycle are divided into two major phases: interphase and the mitotic M phase. During interphase, the cell grows and makes a copy of its DNA. During the mitotic M phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. Interphase. It occurs in three steps: G1 phase, S phase, G2 phase, G1 phase. During G1 phase, also called the first gap phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. S phase. In S phase, the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates a microtubule organizing structure called the centrosome. The centrosomes help separate DNA during M phase. G2 phase. During the second gap phase, or G2 phase, the cell grows more, makes proteins and organelles, and begins to reorganize its contents in preparation for mitosis. G2 phase ends when mitosis begins. M phase. During the mitotic M phase, the cell divides its copied DNA and cytoplasm to make two new cells. M phase involves two distinct division-related processes: mitosis and cytokinesis. Mitosis takes place in four stages: prophase, metaphase, anaphase, and telophase. In cytokinesis, the cytoplasm of the cell is split in two, making two new cells. Importantly, cytokinesis takes place differently in animal and plant cells. Prophase is the first stage of mitosis. In prophase, chromosomes condense and become visible. Spindle fibers emerge from the centrosomes. Nuclear envelope breaks down. Nucleolus disappears. Metaphase is the second step in mitosis. Mitotic spindle is fully developed. Centrosomes are at opposite poles of the cell. Chromosomes are lined up at the metaphase plate. Each sister chromatid is attached to a spindle fiber originating from opposite poles. Anaphase is the third step in mitosis. In anaphase, cohesin proteins binding the sister chromatids together break down. Sister chromatids, now called chromosomes, are pulled toward opposite poles. Non-kinetochore spindle fibers lengthen, elongating the cell. Telophase is the fourth step in mitosis. In telophase, chromosomes arrive at opposite poles and begin to decondense. Nuclear envelope surrounds each set of chromosomes. The mitotic spindle breaks down. Cytokinesis is the final step of mitosis. In cytokinesis, animal cells. A cleavage furrow separates the daughter cells. Plant cells. A cell plate separates the daughter cells.